Hello, we are Erin, Lillian and Remy from the Thomas Hardye School in Dorset, and we are here to present our data on the distribution of Borrelia in ticks in Dorset. So we use genetic techniques to search for the bacteria, which is Borrelia, and this bacteria causes Lyme disease, which is a very nasty disease. So we had to start by purifying the DNA we could find, and we started by, in the summer of 2016, ticks from all around Dorset were collected by dog owners and other animals and given to us kindly. So we started by homogenizing these ticks into small fragments and then heating them and centrifuging them to remove whole cells. Then we centrifuged them again to remove large organelles and then small organelles and extract the DNA in the supernatant. So from there, we got DNA purification kits and filtered out other substances that could be there to make sure that we had pure DNA. And then we moved on to the next bit.
So we first had to determine whether there was any DNA at all in our samples. So we used a mixture of primers and DNA polymerase in order to allow for our samples to be run through an electrophoresis gel. But before any of this, we had to copy out the DNA we had separated millions of times. Otherwise, a single strand wouldn't be visible, as you can see in the bottom right image. Or maybe not from there. But so we did this by using a PCR, or polymerase chain reaction machine, which rapidly copies out the DNA many, many times. To test for DNA with electrophoresis, we first had to make an electrophoresis gel. The gel is made out of agarose powder with a 1 in 50 dilution of TAE buffer, which is then heated to make it fluid enough to mold to the shape of the comb and the container we use. Once we put our gel into the electrophoresis tank, we pipetted our prepared samples into each well of the gel, so the bottom left image, alongside a DNA ladder and positive and negative controls. Due to DNA's overall negative charge as a molecule, we could then run a current through the gel, which is submerged in TAE buffer in the tank, which would go from negative to positive electrodes connected to a power pack. This would therefore separate DNA strands, as said before, due to their overall strand length. After this, our samples alongside the DNA ladder should have gone through the gel sufficiently to be comparable under blue light, which is why you get the image in the bottom right. We carried this out to test first for any DNA present at all in our samples. If DNA was present, then we tested specifically for Borrelia DNA, the bacteria causing Lyme disease. If this bacteria's DNA was present, we could then assume that the tick was infected with the bacteria and that if you'd been bitten by it, you would have contracted Lyme disease.
So we gained no positive results for Borrelia in our experiment, and in the 2017-2018 cohort, figure 1 illustrates our results. So the image reading from left to right, a DNA ladder, a negative control without primer, so no DNA strands were separated out for copying, a positive control, and then all of the consequences results despite being one band being present. This band represents what we think is our primer, giving artifact or false bands where the primer concentration may be too strong due to the wrong dilution. This problem occurred with other years' results too. However, in practice, what should have occurred is different bands corresponding to the positive control that would align with Borrelia if it was present. This is shown in figure 2, where a positive result is obtained as the column label 3 shows a mirror image to the column label 2. As the DNA present was identical to the positive control, in fact, the result is more prominent than the control, potentially due to the fact there are more copies of DNA that have been made.
So overall, we were aiming to find Borrelia in ticks, but however, we didn't find any this year, nor did we find any last year. However, figure 2 is actually from our school, so we have found Borrelia in previous years. Personally, the experience was super fun as I got to develop new techniques, make new friends, and do some extra research that I never would have done without the club. Any questions?
I'm just curious about how the PCR machine, I assume it is, how it actually works. to copy the DNA? Like,
Okay.
what's the mechanism behind it?
Yeah, so just going back, yeah, so that top right image there, so basically that's a PCR machine, and what it contains is, do you know what DNA polymerase is? Yeah, so what it contains is a type of DNA polymerase that has a shorter half-life than normally found in cells, which actually makes it more efficient at copying shorter strands of DNA, which makes it far more efficient at the function we need it to carry out, which is to copy a short strand of DNA. And what happens is we can switch that machine on and it'll just happen over and over and over again until we switch it off or till the set number of copies has been carried out.
Lovely, thank you. Any other questions just now? Yep, one here. Lovely.
What do you think you could do differently if you were to do it again to try and find positive results?
I think first of all would be, well, we got many ticks, so sample size may not be a problem, but if you see on this one, you can't really see it too clearly, but there are bands in every single one, but what those are artifact bands, which shouldn't actually be there. And the only time we didn't get one of those is when there was no primer present. So we're presuming that the primer concentration or dilution was wrong and it causes those and affected all of our results. So in order to go and improve, we could potentially rework our primer concentration, but due to restrictions in school hours and outside of lab time, we can't do that at the moment.
Okay, fantastic. Well, let's just say thanks very much and congratulations again to Thomas Hardye. Thank you.